Hello, in today's video, the last video, I'm going to talk about viruses. First we'll start off with the different types of viruses. We're going to start off with that we have about 3 types. We have the normal virus, which is like the ones like the influenza. They're very small, you can't even see them on a light microscope at all. They're extremely small, and they have a capsid, which is the head of it, and it contains all the DNA and proteins and such. And then they have the tail sheath, which is just their body, that makes sense. And then the tail fibers, which is their arms and hands that can grasp onto. They have receptors that send specific types of cells that will grasp onto it, and then the tail sheath will then syringe its DNA into it. And then we have giant viruses. Giant viruses are kind of the same as normal viruses, except they're the size of bacteria. So imagine a virus being the size of bacteria. I don't know if that seems normal to you, but it's not. Viruses are extremely small and are that deadly at that size. Imagine them being the size of bacteria, like germs and stuff. They can cause serious damage. Luckily, they only infect amoebas, nothing human like us, but you never know, right? And then we also have virophages. Virophages, they're interesting because they need to have a giant virus to even work. So let's say you get infected with a virophage, you're chilling. Nothing's gonna happen to you. You'll be fine. They won't replicate or alter DNA unless a giant virus gets put into the mix, and then they start replicating, activating, assisting with the cell. They'll start assisting because they don't affect other viruses at all. Like, they don't do all that, and they contain genes related to the virus. Like, those virophages have DNA from eukaryotes, prokaryotes, archaea, and like they have other viruses in them, so they can transfer even more diseases into you, which is why they're that dangerous. And now we're going to how they replicate, because just like how we have mitosis, meiosis, they have their own type, and it's called lytic and lysogenesis. Lytic, we're first start off with lytic. Lytic is when they start replicating, like the parts. They start replicating the capsid, the tail sheath, the tail fiber. They start replicating all of that, making copies of it constantly. And then the interesting part is once the cell gets full of all that stuff, it'll start auto assembly. So in a way, it's kind of like, like it naturally, like imagine the force. I don't know, like imagine the force. Like, let's say this pen is this is the virus. It is like all over the cell membrane, and then slowly it's gonna go like that, and all of a sudden it's auto assembled. So it does this but by itself. It already knows where it needs to go, and then it'll cause apoptosis and explode and spread the virus even more. And let's say, oh, that's it, right? No, this is where the lysogenic cycle comes in. Lysogenic cycle is where when the virus first infects the cell, it'll alter its DNA to have the encoding to replicate the virus. So, and then that encoding gets into that cell, starts to cell divide, mitosis. So then there's 2 copies of that DNA that can make the virus. And once the copy is made, then it'll start the lytic cycle. So let's say this one will start the lytic cycle, and this one will do the lysogenic cycle again, and then vice versa, repeat until it is stopped or the host is dead. So let's hope the other one happens. And the difficulty of eliminating the virus is because they have viroids, which are RNA infectious agents that they have no DNA, nothing. They're not even they can't even be touched to begin with. That's how strong they are. Like, they have no protein encoding. They are replicated by cell machinery, and they just help with the growth, and they help the virus grow and such. And then we have pyrons. Pyrons are just enormous proteins that are indestructible because how big they are. They can't be destroyed by regular means, and they're useless, wasting space because they're denatured. So they can't be used. They can't be hurt us, but they hurt us by not being used and blocking stuff because it's just a big nothingness there. And the main key that why something is indestructible is because they can hide in our cell. Let's say so influenza hide it has the capsid head, and that the capsid is hidden in our own cell with our own receptors. So a white cell will see this, like, oh, it's us, but in reality there is the influenza virus inside, but our cells just don't know that until it's too late, and it explodes, and boom, we're infected even more than we were before.